Thank you very much, Professor Lechler, for that kind words and for inviting me to be here at this prestigious occasion. It's a high honor for me, and I'm humbled and very, very, very grateful. I also should give a disclaimer. I'm properly thought of now as a recovering neurosurgeon. I no longer do it. I'm very proud. I've sworn off neurosurgery completely, and so I'm free to talk to you in this room as a physician. What I'd like to do today is summarize briefly our work in understanding mechanisms of bioelectronic medicine. And for those of you who only have time to stay for one slide, if you just remember this encapsulates the entire talk. So bioelectronic medicine represents a convergence of two fields, neuroscience and immunology, that until about 20 years ago rarely intersected in a mechanistic way. But to date back a little, in neuroscience, the idea of reflexes was espoused initially by René Descartes, who, in an apocryphal story, was walking through the gardens at Saint-Germain and saw these robotic mechanized statues that the king had rigged so that when a passerby would step on the sidewalk, hydraulic systems connecting the sidewalk to the statue would make the statue lurch out at the passersby. And the king thought that this was a riot and very entertaining to have the statue scare his guests. Descartes was fascinated by this, and he knew about nerves, and he thought perhaps behavior in animals could also be explained by reflexes, and maybe the nerves were carrying hydraulic impulses like in the robots. And that was the standing theory of reflexes for many, many years until Ramon y Cajal actually discovered and proved neurons. Now, he's well known for showing neurons with the silver stain, but what's been forgotten sometimes is he also showed that nerves propagated information in one direction. And so from that understanding, it became possible to think about sensory information traveling in one way and motor information traveling back out the other way as the fundamental basis of a reflex arc. And this was adopted as a career project by Sir Charles Scott Sherrington, who spent a lifetime delineating the nature of reflex interactions. And a contemporary of his, the father of neurosurgery actually, and the father of endocrinology, was very typical in that he embraced this as a physiological underpinning. And Harvey Cushing showed in his first paper that baroreflex control of blood pressure, heart rate, and respiratory rate moved in response to intracranial pressure. And now the Cushing reflex, which bears his name, which is of course a result of raised intracranial pressure, lowering heart rate and raising blood pressure, he proved that the vagus nerves were an important component of this reflex, because when he cut the vagus nerves, he observed that the correlation between intracranial pressure and blood pressure was even more closely corresponding than when the vagus nerves were intact. So suffice it to say, from that period until then, the 20th century was characterized by physiologists focusing a great deal of work on the role of the vagus nerve as a critical component of the reflex circuits that maintain normal organ physiology. So the vagus nerve here leaves the brainstem and touches almost every organ in the body. It's 80% sensory. There's about 100,000 fibers in the vagus nerve. So some people think of the vagus nerve as a copper wire. It's more like an undersea cable that transmits hundreds of thousands of individual wires to the body's organs, 80% of them sensory. From understanding these circuits to the heart, the blood vessel, the cardiovascular system, the lungs, the pancreas, the GI tract, physiologists have mapped over the 20th century a series of reflexes that account for stable homeostatic organ function that we never think about. And funnily, perhaps, or curiously, the immune system was never on that list. The immune system was left off. And so that was really the state of affairs when I was training to be a neurosurgeon at the New York hospital. And in 1984, I met a patient named Janice who changed my life. She was 11 months old and had crawled across the kitchen floor while her grandmother was cooking pasta. And as grandma turned to drain the boiling water, she tripped and spilled the pot of boiling water on her granddaughter. When I met Janice, she was scalded over 90% of her body, and we took care of her for a month. And although we were literally planning on sending her home from the burn unit at the New York hospital because she had done miraculously well, as we were planning her discharge, one day as I stood in her doorway, she rolled her eyes back in her head and died. And she died in my arms because I took her from the nurse, I gave her mouth to mouth, we worked on her for an hour, and she was gone. And I could not explain to myself or her family or anyone what she died of. She died of shock, but there was no bacteria found in her body. There was no signs of infection. Something had poisoned her cardiovascular system, and we didn't know what it was. And so at that time, 37 years ago, I decided to begin working on this problem, and in some ways, I've not stopped. My colleagues and I at Rockefeller and the New York Hospital identified the molecule that could have killed Janice as TNF. And it wasn't made by bacteria, of course. TNF is a cytokine made by the immune system. But with that knowledge, we were able to do a critical experiment, which was make monoclonal antibodies against TNF and look at the effect of blocking TNF in baboons with shock like Janice had. And what we found in this paper that we wrote in Nature in 1987 was that baboons given infection made a massive amount of TNF, and this killed them. But when we administered monoclonal anti-TNF antibodies to another cohort of baboons that had replicating bacteria in their bloodstream because we'd given them a massive dose of bacteria, these animals were protected. And in fact, if you look at this, it's striking because these are 3 different baboons. This one died of shock during an E. coli infusion. These other 2 have replicating bacteria in their bloodstream, but we gave them monoclonal anti-TNF and they were protected. So this was the first therapeutic description of using monoclonal anti-TNF to target inflammation. And as you heard kindly from Professor Lechler, this has gone on to become drugs like Remicade and Enbrel that are 5% of the world's pharmaceutical market today. Now the model to explain this, I call the cytokine theory of disease. And this has been validated by the use of anti-cytokine therapies targeting TNF, IL-1, and IL-6 in humans, and HMGB1 in animal models. And what cytokine theory says is that cytokines can, in some diseases, be both necessary and sufficient to cause the major signs and symptoms of the disease, from fever to anorexia, fatigue, pain, swelling, loss of function, shock, or tissue injury. Now, it doesn't mean that all cytokines cause all diseases, just like the germ theory of disease doesn't say that all germs are bad. In some situations, cytokines can be necessary and sufficient. Evolution actually retained the activity of cytokines for their health benefits in recruiting cells to injury zones, angiogenesis, antimicrobial activities, dendritic cell maturation, growth factor activities, and the list goes much, much longer. So the key to the cytokine theory of disease is actually how cytokines are regulated. And if you're an engineer, you would put the key regulatory step at the level of cytokine production or cytokine activity. So that's the step that evolution targeted for reflex control of cytokine activity. And in the remainder of my talk, that's the convergence point of neural control of cytokines that we discovered. As a neurosurgeon studying inflammation, you think it would be nice to say that grandma came to me in a dream and we thought it up that way. But that's not what happened. What happened was actually an accident. We had decided in my lab to make other molecules that would target that point in animal models of inflammation. And the molecule that we worked on primarily was called CNI 1493. Now this molecule, as it turned out over 10 years of work, was extremely good at blocking cytokine production by macrophages and monocytes. And we put this molecule in a host of cytokine-mediated diseases, from sepsis to shock, to arthritis, to meningitis, and in all cases, the molecule blocked TNF production and prevented the inflammation. And then one day, we did an experiment, which we collaborated on the report with Professor Pietro Ghezzi, who's here with us today, and we looked at the molecule in the brains of animals with a stroke. And when we put the molecule in the brain, we saw, as expected, that the molecule turned off TNF in the brain, but we didn't expect it would block TNF in the lungs, the liver, the spleen, the gut, and other organs. There was no way to account for this. We had put in a very, very small amount of material into the brain, but somehow the brain was sending a signal to the TNF-producing organs in the body, and it was turning off TNF. And so we puzzled about this for many, many months, and eventually began doing experiments, cutting connections from the brain to the body's organs, reasoning that the neural signals were traveling from the brain to the organs through some unknown path. And when we cut the vagus nerve and put the drug in the brain, now TNF production proceeded as normal. So we knew at that point that there was something about the vagus nerve that was capable of turning off the production of cytokines in the body's organs. And knowing that, it became possible to do the definitive experiment, which was to take an electrode and apply the electrode to the vagus nerve and directly stimulate the nerve and assess the effects of TNF. And so work done by a postdoc, Ludmila Borovikova, then at the time in the lab, and published in Nature, show that what happens. If you give endotoxin or other stimulating agents to a mouse or rat, the animal will overproduce TNF. If you cut the vagus nerves, they make even more. So it's like cutting the brakes in your car going down the hill. You've lost the controls. When we electrically stimulate the vagus nerve, the TNF production is blocked. And because you're blocking the molecule that causes shock, you can measure blood pressure as well. Here's animals going into shock. They go into shock sooner if you cut the vagus nerve. But if you electrically stimulate the vagus nerve, block the TNF response, the animals are protected. So this was incredibly exciting. This was in the late 1990s. I sat down at lunch one day on the back of a napkin, and I wrote out an idea of building a nerve stimulator to implant on the vagus nerve in patients with rheumatoid arthritis or Crohn's disease, turning it on, blocking their TNF like the monoclonal antibody does, and treating their problem. But of course, you can't do that. You're going to have a whole session on clinical translation. You can't just go from an idea to clinical trials. You have to understand mechanism. And so my colleagues and I spent many, many years mapping out the mechanism. And what we learned, which was extraordinarily helpful in this process early on, was that in animal models of endotoxemia, most of the TNF is made in the spleen. And so this helped us because we were able to now study this problem of neural signals arising in the brain, going down the vagus nerve to the splenic nerve, perfusing the spleen as a neural network, and how could this control TNF? And what we discovered is that if you look inside the spleen, you can actually see the cells in red here by immunohistochemistry that are making the TNF. So this is work done by a former grad student in the lab, Mauricio Rosa-Spallina. The cells in red are making TNF in A and B. They are mostly red pulp macrophages and marginal zone macrophages. If you electrically stimulate the vagus nerve or the splenic nerve, you can see in C and D that the production of, oh, you can't see. Well, A and B has a lot of red dots that you can't see. But C and D is essentially the red dots are turned off. The point is you can electrically stimulate in the brainstem and follow the signal into the spleen and look at the individual cells that are making the TNF. And that allowed us to now ask the next question: how does the signal get from the nerve endings to the macrophages? Well, there came another surprise. It turns out, in work done by Mauricio and Peter Olofsson, who's here, a professor from the Karolinska now, who I've had the privilege of collaborating with for many years, Peter and Mauricio discovered that the neural signals coming down as action potentials in this orange fiber here converge on cells that are green. In this case, they're green because they're transgenic T cells that are making acetylcholine. So this is, it turns out to be a small subset of T cells in the spleen and other immune organs that express choline acetyltransferase, the rate-limiting enzyme in the biosynthesis of acetylcholine. So this is a chance where the neural signal enters the spleen, releases norepinephrine, the norepinephrine communicates with these T cells, the T cells make acetylcholine, and acetylcholine, as you'll see in a few minutes, turns off the TNF response. So this was incredibly exciting. To prove the role of these T cells, Peter and Mauricio studied nude mice, which don't have T cells. And when you electrically stimulate those animals, you can see that TNF, if anything increases, it does not decrease, as opposed to the wild type animals that have T cells. Now, really interestingly, you can take these mice and put those green T cells that make acetylcholine back into the nude mice and repeat the experiment. And when you do that, you restore the phenotype. So this is proof here that the vagus nerve signals enter the spleen, control these green T cells, I call them green T cells, these CHAP-positive T cells that make acetylcholine. So Peter became very interested in these T cells because they represent, to our knowledge at the time, an unknown subset of the lymphocytes in the spleen and other organs, as I said. So Peter studied these, and what he found was striking. Now, this paper was published a year ago in Nature Biotechnology, and I checked this morning. It's still number 8 on the impact factor list for all of Nature Biotechnology in the last year. It's a very important paper, and kudos to Peter for this. What Peter shows in this paper is that analysis of these T cells against 198 different well-described immune cell subsets in the ImGen data set shows that they're unique. This green bar is the expression of choline acetyltransferase. If you cluster them with other T cell subsets, you see that they cluster as CD44 high, CD62 ligand low regulatory T cells here, clustering with the CD4 positive and the CD4 memory cells. If you look at pairwise Euclidean distance plots of the complete gene expression, it turns out that these T lymphocytes are highly segregated. So putting those last 3 sides together, that allows us to compare these cells against 13 lymphocyte subsets, 41 T cell transcription factors that are highly over or underrepresented, and allows us to conclude that we can call these cells by a unique name, which we call CD4 T-chat. And for those of you interested in either T lymphocytes or the control of blood pressure, I urge you to read this paper because we found in that Nature Biotechnology study that these cells have a critical role in maintaining resting blood pressure because they deliver acetylcholine, which is the stimulus to EDRF, to resistance blood vessels. So that's where we are in terms of following the electrical signal to the T cell in the spleen. The T cell makes the acetylcholine, but then what happens? So this was a really interesting problem. We had pharmacological evidence that the signal was nicotinic, a nicotinic signal to control the cytokine release in the macrophages, but we didn't know which receptor. We knew that nicotinic receptors are pentameric ligand-gated ion channels. We knew that they're comprised of 10 different alpha subsets and 4 different beta subsets as homopentamers, things like alpha 2, alpha 3, alpha 5, beta 4, or alpha 3, beta 4, or as a homopentamer called alpha 7. We didn't know which of these was involved, and so when Hong Wang began to research this problem to understand which of these receptors could be participating in the signal that would turn off the cytokines downstream of these T cells, Hong and I and all of us were very surprised when it turned out to be a single receptor, the alpha 7 homopentamer. And so in a paper in Nature in 2003, you can see here wild-type mice given vagus nerve stimulation, the TNF production is blocked. The alpha 7 knockout, which are of course identical to these mice other than the deletion of the alpha 7 gene, overexpress TNF compared to the wild type, and the vagus nerve stimulation fails to decrease. So like cutting the vagus nerve, cutting the splenic nerve, taking out the T cells, or knocking out the alpha 7 receptor, all of those things converge on a pathway which we called the inflammatory reflex, reasoning that inflammatory signals would travel up the vagus nerve and activate a motor path that would counter-regulate through this downstream path to the spleen the overproduction of inflammation. So that's what we were doing in New York, and not well known to us, but now very well known to us, was investigations going on in Amsterdam and later in the UK, led by a member of your society, Paul Peter Tak. Before I get to that, sorry, I forgot one more slide. I won't go through the detail, but after alpha 7 binds acetylcholine, the effect on the mitochondrial membrane, where the alpha 7 expression is predominant in macrophages, is to stabilize the mitochondria, downregulate the release of mitochondrial DNA, prevent the inflammasome complex from activating cytokines like IL-1, IL-18, and HMGB1, and separately prevent the nuclear translocation of NF-kappa B. The point of the story in excruciating detail is that we understand the mechanism, from applying action potentials to the vagus nerve down to the level of gene transcription and inflammasome activation. But as I was saying, Paul Peter Tak was working independently And he was screening fibroblast-like synoviocytes from rheumatoid arthritis patients using 2,343 adenoviral shRNAs. Now, he was screening these cells in the presence of TNF, and he was recording the production of IL-6 and IL-8, and he was looking for viral transcripts that would knock down the production or increase the production. And what he discovered, the transcript that kept coming up was the alpha-7 nicotinic acetylcholine receptor. He looked up and found our work. We began speaking. He continued to work independently and collaboratively and showed that this nicotinic acetylcholine receptor on the fibroblast-like synoviocytes downregulates cytokines. So here you see TNF is stimulating IL-6, TNF is stimulating IL-8. When he titrated in ARR 17779, which is a highly selective alpha-7 agonist, you see this dose-dependent inhibition of IL-6 and IL-8. Paul Peter and his colleagues had discovered that alpha-7 is expressed in these fibroblast-like synoviocytes, and that the alpha-7 expression is critically important to downregulating the production of cytokines, much like I've been telling you the alpha-7 can downregulate the production of cytokines in the spleen. He then went on to show, excuse me, that the alpha-7 knockout mice have much more severe arthritis. So here's arthritis in the wild type, and here's arthritis in the alpha-7 knockout mice, and that their radiologic scores are much worse. He then went on to show that if you treat these arthritic animals in wild type mice with either saline or ARR 1779, that you get a highly significant protection against the joint destruction that you see here. This is a significantly improved histologic picture, and this is histologically scored here. So a therapeutic benefit from ARR 1779. And finally, in collaboration with a company that I co-founded called Setpoint Medical, in order to begin to do clinical trials of this idea in patients with rheumatoid arthritis, Yakov Levine at Setpoint and Paul Peter Tack as the consultant, as am I to Setpoint, showed that if you implant rats with a vagus nerve stimulating device and turn it on, that you confer some protection against arthritis. So these are animals that have an implant that's not turned on, and they get severe arthritis. And here's the attenuation of the arthritis by turning on the electricity in the device. And again, histological protection against joint damage with preservation of the cartilage and less bony destruction, as you see here, attenuated significantly here. So at this point, we understood the mechanism. The signals travel to the spleen, where they converge on neurons that release the first neurotransmitter, norepinephrine. That regulates T cells that release the second neurotransmitter, acetylcholine. The acetylcholine travels to the macrophages that express alpha-7, and the interaction of acetylcholine with alpha-7 turns off the production of TNF, IL-1, IL-6, IL-8, and HMGB1, and it actually turns on the production of IL-10 and some other anti-inflammatory mediators. So now we were ready to do a clinical trial. Now we understood the mechanism, and fortunately, we had Paul Peter Tack as a collaborator to lead the clinical trial. The trial was done here in Europe at centers in Amsterdam, Bosnia, and Croatia, and there were two cohorts of patients, patients who'd failed methotrexate, primarily in Bosnia and Croatia, and patients who'd failed methotrexate and multiple biologicals, primarily in Amsterdam. The patients were implanted with a device much like a cardiac pacemaker that sits under the left collarbone, and then a lead was tunneled up to their left vagus nerve. The device was turned on for a period of time and then turned off because we couldn't do this first trial as a placebo-controlled trial. So by turning off the device, we could get an indicator of the role of the electricity being on or off. And you can see this device was manufactured by Cyberonics. It's marketed for the treatment of epilepsy. More than 200,000 patients have been safely implanted with this device in the last 25 years. But we reprogrammed it because it turns out, and it's a much longer conversation than today allows, unless there's questions afterwards, it turns out that the charge parameters that require activation of a small subset of fibers in the vagus nerve, we think less than 1,000, is on the order of 400 to 500 microamps, which is a very low level of charge. The resting charge requirements for epilepsy patients is a lot more than that. So by cuffing the entire nerve but setting the charge at 400 microamps, we're still able to garner a level of specificity to the fibers that go to the spleen without affecting heart rate or other GI functions or other functions that could theoretically be side effects. We did not see serious side effects. The first part of the experiment was to enroll patients during general anesthesia to get another insight into the possibility of a placebo effect. You see here anesthetized patients produced TNF from whole blood monocyte assays before and after anesthesia, but when the device is implanted and turned on, there's a significant attenuation of TNF production. In the same patients during anesthesia, while they're still asleep, there's also a significant attenuation of IL-6 and IL-1 beta. So now we know that the inflammatory reflex mechanisms that we've observed for 20 years in animal models appear to be preserved in humans, and it was time to move to the clinical study of the clinical outcome of the patients with RA, which we reported in the PNAS last July. This paper is also in the top 20 citations for the journal out of 250,000 papers in the last year. What happened in these patients was striking. There were two cohorts, cohort 1, cohort 2, methotrexate failures, biologic failures, and also showing you here the combined in the middle. This is the DAS28 CRP score, which combines clinical endpoints like pain and swollen joint counts done by standard rheumatology practice, as well as serum CRP markers, which of course are objective measures. As you can see, the patients were implanted, the device was turned on, they developed highly significant improvement in their clinical scores until the device was turned off, and then they worsened. And then it was turned back on again, and they continued to improve. The results of the follow-up study is coming up to, we have 3, 4, and 5-year follow-ups from many of these patients, and the results are not yet published, but several of the patients have remained in complete remission. Here's the correlation, if you will, of the TNF inhibition to the clinical response. So the TNF is plotted on the right, and it's these squares, the upper line here, the lower line here, and the clinical score is plotted on the left, and it's the circles. So there's a decent correlation between the effect of the treatment on the clinical score and on the biomarker of whole blood TNF production, which was the target of the device. This is the first patient. He's in Bosnia, in Mostar, Bosnia. This is his physician. She was very shy. This is her boss, the big department chair. He was very proud. This is the electrophysiologist. And I don't know if the patient looks happier or I look happier, but we were both very happy. Now what was amazing is I asked this chairman, he would not speak English to me, although he understood every word I said. He was shy. And I said to him, I finally got him alone in the corner, and I said this, because we had set up his hospital with a little laboratory to do the experiments and obviously funded the trial. And I said to him, this trial, did it work? He goes, I said, you didn't think it was going to work, did you? He goes, and that's an important story for the Q&A. If you don't understand the mechanism to these devices, they can be perceived as snake oil. But that's the opportunity we have, because the story I just told you about TNF can be applied to your favorite molecular target or your favorite cellular target. Pick your target, pick your disease, and rather than screen for molecules, now we have the opportunity to screen for neural circuits that might normally regulate that target in a homeostatic way and to develop new devices to control those targets. That's called bioelectronic medicine. There's another case that was reported in the New York Times. This case is remarkable from the same study because she was 38 years old, 16 years disabled by rheumatoid arthritis, had failed methotrexate, failed 5 biologics, and on some days couldn't pick up a pencil or button her shirt to go to work in the morning. And at last report, since April of 2013 when she was implanted, she's now riding her bicycle 20 miles round trip to the Dutch coast on weekends. She has remained in remission. So it's incredibly exciting time to think about building devices, but it's also an incredibly exciting time to think about what this means physiologically. Paul Peter Tack and his colleagues recently reported that this may not just be a treatment in window, but there may also be some diagnostic implications as well. It's been known for decades that vagus nerve activity is depressed in patients with lupus, rheumatoid arthritis, inflammatory bowel disease, sepsis, and other inflammatory disorders. But it was always diabetes, but it was always presumed that the nerve damage was downstream of the inflammation. Paul Peter took a different approach, and he actually raised the hypothesis that the nerve damage might precede the onset of the inflammation, because as I said before, it's like brake failure going down a mountain. If the vagus nerve system has become impaired, maybe that allows the overexpression of inflammation. And so he studied healthy subjects, individuals at risk of developing RA. These are patients with either genetic family history or serum markers of potential RA with no signs of rheumatoid arthritis, and RA patients. And he looked at resting heart rate and found that individuals at risk had significantly higher resting heart rate, which was similar to the resting heart rate in rheumatoid arthritis patients. He then did a validation cohort of 50 subjects and showed that in the patients in whom no arthritis developed, arthritis went on to develop in patients whose heart rate was higher. So this is a fascinating way of thinking a little differently about diseases that we all have thought about for many, many years and know a lot. It might actually be that these reflex signals that we can usurp or harness as therapeutic insights may also contribute to the disease pathogenesis if they become impaired. And there's a lot of evidence for their impairment. So this is where we've come. We've come to an understanding. I didn't talk a lot about it today. We have a good knowledge about how infectious products or sterile products like HMGB1, sterile products of cellular injury, can interact with toll receptors and with cytosolic receptors like NLR, RIGs, nods, and inflammasomes, and that these culminate on the production of cytokines. The cytokines are actually sensed by the vagus nerve. I don't have time to talk about it today, but the vagus nerve is 80% sensory, and we and others have evidence that the sensory interaction of the vagus nerve with inflammation sends signals to the brainstem that activate the outflow. The outflow travels down the vagus nerve to the splenic nerve in the case of the inflammatory reflex. In the case of other reflexes, the motor signals can culminate on lumbar spinal cord roots in multiple sclerosis. They can culminate on the spleen in antigen antibody vaccination responses. They can culminate on the adrenal gland in the case of some models of sepsis. They can culminate on the mobilization of cells from the bone marrow, and they can culminate actually on signals in some cancers where inflammation has an important role. So the opportunity to understand the inflammatory reflex has been significant, but the opportunity to map new reflexes that will have new implications for other targets and other devices is extraordinary. That process of going from molecular target to neural circuits to device development is a process of bioelectronic medicine. We at the Feinstein Institute in New York are making major investments as a not-for-profit research center to be the home for bioelectronic medicine for the world in New York. And we are working closely with Peter Olofsson and his colleagues at the Karolinska Institute, where he's leading a similar effort there to build a major research home for bioelectronic medicine. The opportunity for young people to come into this is extraordinary. The opportunity for old people who've thought about targets for many, many years and want a new way of thinking about it to do this is extraordinary. And the opportunity for companies, startups, big and small, to come into this space and build devices in which the specs of the device are dictated by the needs of the neural circuit to control the target is extraordinary. As recently as 2 weeks ago, I was speaking with Francis Collins about his and the NIH's commitment to what he calls SPARCs, stimulating peripheral nerve activity to relieve conditions. Modulation of nerve signals to control organ function has therefore been recognized as a potentially powerful way to treat many diseases and conditions, such as hypertension, heart failure, gastrointestinal disorder, type 2 diabetes, and inflammatory disorders. It's remarkable. He has committed $250 or $300 million to this, and hopefully more to follow. There's been major investments here in the UK and across the world by GSK, led by Moncef Slaoui, Chris Pham, and Paul Peter Tak. They have together with Google, collaborated on a new company called Galvani, which is committed to developing devices in this space. SetPoint has been supported by Boston Scientific. DARPA has supported my lab. The NIH has supported my lab. Covidien has supported SetPoint. GE has done a major deal with the Feinstein Institute. This is a burgeoning area worth hundreds and hundreds of millions of dollars. The investment into Galvani alone was estimated, according to the business pages, $750 million. So we're looking forward now to one last take-home point. I don't want you to leave the room thinking that this is all about the vagus nerve. It's all about a device. The vagus nerve is a copper wire. All of those are wrong. The vagus nerve is 100,000 fibers. The devices can be highly specific, and we are at the tip of the iceberg for mapping new circuits. This is a paper that summarizes some of these advances. We just published this a month ago, my colleague, Sangeeta Sivan and I. But there are neural circuits that have been mapped to the bowel wall, controlling the differentiation of macrophages, to the adrenal gland I mentioned, to the spleen. I mentioned the importance in vaccinations, same for lymph nodes. Axon-axon reflexes are sensory reflexes in which sensory nerves in your soft tissue interact directly with the presence of inflammatory stimuli or bacteria, directly. So the nerves are on the front line. It's not just neutrophils and macrophages. The nerves can sense the presence of bacterial products. That information is nearly instantaneously transmitted into the central nervous system and back to the tissue through axon-axon arcs. It has important implications as we're learning on immunity, and this is work led by Cliff Wolf at Harvard. And finally, similar processes are occurring in asthma in the lung, led by Clifford's protege, Isaac Chu, who stayed at Harvard. He wouldn't come to New York with me, so be that. So fantastic work. And finally, the last story is a Cell paper from Professor Arima's lab in Japan, in which he studied the entry of pathogenic T cells into the central nervous system. He hung mice by their tails in their cage. So now the mice are only able to walk around the cage on their front legs. And what he observed is that when he administered these pathogenic T cells into these mice, is they couldn't breach the blood-brain barrier at the lumbar spinal cord. And what he figured out was that the neural signals that were fed by the activity of the muscles, as in response to muscle activity, sensory signals, were activating a reflex that opened and closed the blood-brain barrier right at that place. And so he mapped with molecular precision a mechanism through CL20 that opens and closes the blood-brain barrier. He mapped a neural circuit that allows pathogenic T cells to get into the blood-brain barrier. And when he put the mice back on all four legs, they got multiple sclerosis in their spinal cord. So that's a fantastic paper for those of you interested in multiple sclerosis. This is the tip of the iceberg. There will be, I estimate there will be dozens more discrete neural circuits controlling discrete immune functions in the next few years that will be reported by many labs working on this. We also have more powerful tools now than ever before. My colleague, Valentin Pavlov at the Feinstein, is using optogenetics to specifically target neurons in the basal forebrain cholinergic system. Mice have been here genetically engineered to express a channel rhodopsin, YFP channel rhodopsin 2, in cells that express acetylcholine. These cells here in yellow, when you shine a light on them, will fire, and we know that they're cholinergic neurons. When you fire the light on these neurons in the basal forebrain of these mice, and you look in the periphery, you see that the non-carriers make TNF, and the light on in the carriers block the TNF in the spleen. This means that a few neurons in the base of the brain can be selectively controlled, in this case with channel rhodopsin through optogenetics, but in the future through selective implants, to target those brain neurons to control the immune system in the spleen. It's really remarkable. So in closing, I'll go back to what I said a minute ago, that there's opportunities here for investigators to think about drug targets in a new way, and I would encourage the society, the academy, to think and strongly support additional research investments in the basic science necessary at the convergence of neuroscience, molecular medicine, and biomedical engineering, because this is an extraordinary opportunity for our patients. Thank you.